Hey guys, welcome back. I'm Siddharth from Electro and today we'll be talking about CRISPR. So let's at least know what it is. So it's a topic related to biology, and CRISPR is a defense mechanism used by E. coli. It's a bacteria to fight against viruses. So why is it so interesting? So it's so interesting because we have harnessed this defense mechanism for our advantage to edit genes. So in this video I'll be discussing how this CRISPR-Cas9 system works, and other than that we'll be discussing some applications of the same. So let's begin. First off, what does CRISPR stands for? So CRISPR stands for clustered regularly interspaced short palindromic repeats. It's a mouthful, but let's break it down. First, that it's used for gene editing. So this CRISPR system, this Cas9 system, is found in a specific bacteria. Its name is E. coli. Now, to understand this whole thing, let's first talk about how we fight diseases. So it will give you a good idea of what's going on here. So when we are affected by any disease, what our body does is that after fighting the disease, if we fought the disease successfully, the body will keep the antibody used to fight the specific disease. So the next time the disease tries to affect you, the body can automatically develop those antibodies as it has a copy. So as soon as that happens, the body will start generating new antibodies to fight that specific disease. So it's easier for the body to do that. Now, a similar process takes place inside of E. coli, the bacteria in which this system works. So the first thing is that let's first break down the whole acronym, that's CRISPR. So it says clustered regularly interspaced short palindromic repeats. So palindromes are words like you have the word Malayalam, so you speak it from left to right or right to left, it's one and the same. So these are simply sequences of base pairs which have palindromic repeats. Yes, so suppose you have an ATGC sequence. So on the top with the helix, on the other helix you'll have the same sequence. So it's a palindromic sequence. So those palindromic sequences can be seen here. They can be seen here, here, and here. So you see the yellow ones, and then you have these spaces in between. So they are clustered regularly interspaced. So they are clustered together and they're regularly interspaced, and they are short palindromic repeats. So these can be from 30 to 100 characters short or long. Now, why is this? How is this used? So this is the genome of that specific bacteria you see here. So you have some Cas genes associated with the CRISPR system. So Cas genes are CRISPR associated genes. Now, what do these genes do is that they synthesize a protein called Cas9. So this protein, we'll talk about this protein in detail in just a minute. So now, for example, here's a bacteria, here's a virus which affected the bacteria E. coli. So once it's done that, what it will do is to infect the bacteria. It will send an DNA inside the body of the specific bacteria, that's E. coli in our case. So when it does that, the DNA of that virus will start affecting the bacteria. It will try to hijack, it will try to use the machinery of the cell, and if it does that, what will happen is eventually the bacteria will die, and the virus will easily win the fight. And what happens is, if in a bacteria, if it's able to fight the disease properly, then what it will do is it will keep a copy of that DNA that was trying to infect the bacteria in the first place. So if the same thing happens the next time, the bacteria E. coli can easily use that specific DNA of that specific virus to fight against it the next time. So once it's fought the virus, what it will do is it has these It'll keep the DNA of that virus inside these in between these palindromic repeats. So the repeats are here, and the interesting place is where the spacer sequences are. So these DNA of these specific viruses, they will be kept in between these palindromic repeats. So now what happens after that? So for example, so the next time if say the virus affected the bacteria, what will happen is it will make an RNA. So suppose this was that spacer DNA we had here, the spacer DNA of the virus. So it will be unwound by an enzyme called helicase. So once it's done the unwinding, it will make a guide RNA, a tracer RNA. So what will happen is, see, you have these base pairs lying around here. So it will make an RNA. So you see, once the RNA is made, it will be of the similar. It will contain the similar base pairs. So for T it had A here, and for G it had C here. So for T it will have A here, and for G it will have C here, and that will be done by a procedure called transcription. So you see here, the helicase will unwind the DNA, and these DNA polymerase they will. So, this is actually not a photograph of making guide RNA, but a similar thing goes on. So when it's open, here a guide RNA is manufactured, so it will have the same sequence as the DNA will have. So once that's done, the tracer RNA will be sent to the Cas9 protein, which was synthesized by the Cas genes. So these are so once that DNA is sent to Cas9, just a second. So once that DNA is sent to Cas9, you'll see that once that RNA is sent to Cas9, once it's reached to the Cas9 protein, another RNA will be generated to check whether or not it matches the sequence of the target DNA. So the target DNA is the DNA of the specific virus which is affecting the bacteria. So if the sequence matches, what will happen is the Cas9 protein, it has nucleases. So nucleases are enzymes which cut the phosphoric bonds of the double helix. So if it does that, then you can see those strands will be cut, and this is pretty much how the CRISPR-Cas9 system works. So so the question is, how will we achieve gene editing using this technology? So this will be done by a pretty simple way. So once so what we can do to edit genes is that instead of the tracer RNA that's being it's a tracer, or say the guide RNA, which is being generated from the DNA of the virus, instead what we could do is send a specific RNA which we have generated to the Cas9 protein and give the target DNA of the DNA we want to edit. So for example, we sent a tracer RNA which has some amount of nucleotides, some number of nucleotides, so that RNA will contain the information of where to cut. So once we have sent it to the Cas9 protein, the Cas9 protein will cut the DNA, the target DNA which was sent by us to it. So once it's cut, the whole cell is in a chaos. The cell wants to rejoin or say repair the DNA. So instead of it can do two things. It can either just join the two ends which were broken together, or else what it can do is use other nucleotides lying around in the cell to join to match the sequence and rejoin the whole the double helix. So that they are the two things that can be done. So instead what we can do is we can put a DNA close to the Cas9 protein. So as soon as the target DNA is cut, that specific gene, the DNA we want, that will come in place of where the DNA was cut. So this way you can see the DNA was cut at a very precise place, and at the same time we decided where it was cut, and we decided what gene was replaced at what place. So now you see, using this we could do a lot of things. So the first application would be designer babies. So you see, all of the human characters like your hair color, your eye color, then your skin color, and there are a lot of other things that are controlled. In fact, your whole body, all the traits you have, they are all controlled by specific genes. So if we could edit genes, we could pretty much change the organism itself. So here, if you have a human embryo, what you could do is use the system to replace specific genes. So you could literally sit down with your doctor and talk about, yeah, I want to change my child's hair color, I want it to be blue or green or anything you want. Pretty much you have control on what your baby will look like, what will his IQ, her IQ will be. So that's a great application. And the second is we could be immune to most of the diseases, as we could literally target each and every cell. We could not target each and every cell, but we could target DNA which is affecting the human body. So for example, we had some bacteria which was affecting the whole body. What we could do is use the Crispr system such that it would find those bacteria. It will check the DNA, and if it does match with the DNA we provided it with, then it will cut the DNA, and then pretty much the bacteria will die. And the next one is quite interesting. It's called anti-aging. So most of us think that aging is something that happens because cells start deteriorating in number, or say they don't work properly, and the mechanism responsible for repairing them doesn't work properly. But that's not totally true, because there are some genes specifically which are specifically directly associated with aging. So those genes cause aging simply. So if we can use this technology to change those genes, and or say deactivate those genes, what we could do is that we could either stop or reverse aging. So that's another application. So I hope you enjoyed the video, and if you liked it, please give it a thumbs up and comment down below. I'd like to see what you think about this technology, and if you have some other applications, do write it down in the comments below. The people could see it. And if you have any questions, you can ask, and don't expect really good answers from me, because I am not an expert in this thing. So bye for now.